Hi, my name is Itai Ben Porath, and we're now at the tissue culture facility, tissue culture room. And this is where we grow cancer cells that come from patients, mostly breast cancer cells, but also other types of cancer cells. Some of them have been grown in Petri dishes for years and years, and some of them are just out of the patients. And what we mostly do is either put in genes or take out genes. And then we test the effects of these particular genes and how these tumor cells later behave. Are they now more aggressive? Do they form tumors more easily? Or now does taking out a particular gene really stop tumor growth? And this is what we're really interested in. So I'm going to take out one of those dishes and maybe we can take a look at them under the microscope. And these are breast cancer cells that were extracted or taken out of a patient many years ago. And we're very interested to see what makes them aggressive. These are very aggressive cells and they're very metastatic. So we're really trying to figure out what genes make them so. Okay, so we're now in my main lab. Welcome. This is the Central Research Building in the Medical Institute here at the Hebrew University. And this is our microscope room. And let's look at these cancer cells under the microscope. Okay, so now we have these breast cancer cells here under the scope. And as I said, we're really trying to manipulate them in all different ways genetically, putting genes in and out and seeing how they grow in the dish, and then later how they will be able to form tumors. And one thing that is very useful that we do is we put in a gene that comes from a fluorescent jellyfish. And this gene turns the cancer cells into green cancer cells, green fluorescent cells. And actually, you might be able to see on the screen that each one of those green spots is an individual cancer cell. And this now really assists us in following and tracing these cells when we look at them in the living animal, which is the mouse. So in later experiments, when we're asking, are these cells now aggressive? Can they metastasize? We're now able to trace these green cancer cells, say, in different organs in the mouse. So for example, here's an image of a lung that came from a mouse, and you can see that it's all spotted with these green cancer cells. So this now indicates that these tumor cells are able to reach the lungs and they're able to settle there. And now the question is which genes really help this trait of the cancer cells and which genes we can turn off to stop this process. So thanks for visiting us, and it was a great opportunity to share some of our work with you, and come back again in a few years. We'll probably be much further ahead. We'd be glad to share it with you.
Itai, it's really good to see the lab work, because I know you can be working 2 years to get this understanding of one little mechanism between one gene and one chemical. It's a long process.
Absolutely.
But we can summarize it up. I want to go into the subject of metastases,
right,
because we all know cancers multiply. In the breast cancer, you get multiplication, you get breast cancer.
Right.
But the real problem comes when they get out of the breast. Now, it's really difficult to figure out. The cell has to get out of the breast, get into the bloodstream, travel down the bloodstream to some other place, get out of the blood vessel, cross some rough territory, get into a liver where it doesn't belong, and why doesn't the liver kick it out and grab it and, macrophage surround it and destroy it? Why does that happen? How does metastases work?
Well, it's really a very central and confounding question, because it really is an amazing biological feat, just as you're describing. And there's a major question in the field: A, how does it happen? And B, why does it happen? Because it's not immediately obvious that it's self-serving for the tumor cells to do this. They might be happy just where they are. And in fact, it's been, it took about 2-3 decades to figure out the basics of why normal cells, or how normal cells become tumor cells and how they form this, primary, the first solid mass of a tumor. And only really in this last decade, scientists were able to start scratching at this very, very complex process. It could be that every single step on the way, something changes. Maybe there are genes that control how the cell leaves the tumor, how it enters the blood vessels, and there are genes that have been shown to be able to do that. And it could be that there's another gene, such as MYC, that's kind of a master orchestrator that changes the whole nature of the tumor cell and now allows it to do this. And there
so that would be the Satan in charge of the whole process?
Yes, but again, the question is, what motivates the Satan? Maybe it's trying to do something completely different, and this is just a byproduct of another completely different process. Maybe it's in the nature of the cell where the tumor started. So we know that melanocytes, either the pigment cells in the skin, these cells are naturally very motile. In development, they move. And when a patient gets melanoma, which originates in one of these cells, these tumors are very, very metastatic. The tumor cells are very motile as well. And why? Because they've just inherited this trait from the melanocyte, whereas a keratinocyte, another skin cell, which is used to sitting in its place and not going anywhere, will cause a benign tumor that stays in the skin. These are basal cell carcinomas, squamous cell carcinomas. It can be easily removed in an operation. So it could be inherited by the nature of the cell itself.
Now, when you talk about these cells, I picture The Great Escape or some prisoner movie,
right?
Because it sounds like that, it
Breaking
reads like
free.
Yeah, that's right. But one of the things, I read your paper, by the way. It's a very difficult paper. It's the most difficult one I've read in this whole series. The one in 2008. It's got quite a bit of
computational analysis. That's the problem. Yeah.
It was a hard read, but I get the sense that you're onto a whole new thing here, which is the link between cancer and stem cells.
Correct.
That, I thought stem cells were all good things because they allow you to, we'll get regenerated organs, we'll get all kinds of good things. And it turns out they may be that we're getting cancer out of it. Is that right? Is that the story?
That's the exact way to put the link, and it is a fascinating link, because here we have these normal stem cells that are good. They contribute to maintaining our tissues and generating them in the first place, and that's through their capacity to, it's called self-renewal. They maintain their identity. They don't senesce easily, so they can, throughout your lifetime, they can stay fresh and young. But if you think about it, that's exactly what the cancer wants to do. So if we look at what makes a stem cell always young and able to self-renew, which genes control it, which master genes, such as MYC and others. Actually, MYC is involved in this process.
Oh, the same one.
The same MYC, it turns out, also is important for stem cell function. So which genes in the stem cells provide this trait? And then what we did in this paper you're describing was actually very simple. We said, let's look at the genes active in the stem cells and ask which of those is also active in tumors. And it turns out they are active, some of these master genes type of regulators. And they're active not in every type of tumor. You can see that in the aggressive tumors.
Like a blastoma.
Glioblastoma, exactly, aggressive brain tumors and breast cancers. The more aggressive the tumor is, the more you see the activity of these stem cell related genes. And in fact, a lot of what we're doing now is trying to see the contribution of each of these factors. And we're looking specifically for those master type of genes, the ones that really program the cell. Which of those are active in aggressive breast cancers, and what is the contribution of each of those? And we found several factors that really can change the nature of the cancer cell and turn it to be more stem cell-like.
Oh, and not get into really bad trouble. And
right.
You can reform them.
Well, yeah, that would be the next question. How do you stop this?
But can you do, at this stage, can you do diagnosis to tell when it's going to be a good stem cell and when it's going to get bad and go wrong?
I mean, that's
like doing, which guy's going to become the criminal and which is going to be the good guy?
No, exactly. Once you identify such a candidate, one of the first things you do is you go to the clinic and you look at those cancer specimens, those samples, and you ask, well, in which specimens this gene is active in and which others is it not active, and Can we show that the outcome of the patient differs between the two groups? So that's a diagnostic tool. And then, of course, you're asking, well,
Can we do that now?
To look at...
is it diagnostically useful right now, or is that a future
We're
prospect?
exactly, our factors, we're exactly at the stage now where we're looking at collections of hundreds of patients that we're treated at this medical center and testing the activity of these different factors and trying to assess the contribution. We're really right now trying to do this.
So you may be on the verge of a new breakthrough in diagnostics.
Hopefully, hopefully.
And then what about therapy? When are you going to learn to turn the bad guys into good stem cells?
So that's the next step. You would like to say, let's hit this guy on the head and silence its activity. Of course, that turns out to be very, very challenging. I mean, once we do the basic research, and even if we get to a pivotal understanding, here's the guy, this is contribution. Now, on average, it takes, best case, about 15 years to figure out, if we can at all, how to stop it, how to stop this guy. And then you take it to the clinic. Sometimes it works. There's been amazing successes in the past few years, but often it doesn't work, and you get surprising different effects. The drug that you thought will stop the tumor in one way, it turns out the tumor comes back and activates a different gene, and now things are even worse, or it just finds a way to overcome it very quickly. So this is a major, major challenge, the distance between understanding what happens and actually stopping the tumor. There's a great length of time and resources there, but there's amazing advances that are just coming into the clinic in these years.
Well, that's the sense I've got out of this series of interviews, that you're on the verge of big breakthroughs, not just minor breakthroughs or specific, and it has general applicability, because they're not just about, like you're working on breast cancer, I gather,
Right.
but it's about all cancers, I assume. They're very similar in that kind of mechanism, though there may be different genes operating, etc.
Yeah, absolutely.
Do you ever get tempted to get into the clinical side of it, or are you quite happy in your lab?
I think for us, one of the major attractions is really to understand the basic of how things work, what motivates the cancer cell. There's so much that we don't know, that even at the most simplistic level, that for us, it's really the joy, the challenge is to figure out how things work. But then, of course, there's a handoff. How do you translate that into a possible drug? We're actually, that's, of course, an essential field. We're less trained to do that. And then at the last stage, once you have the drug, then you have the years of testing it. That's the third stage. So each one of these stages is very challenging and requires a whole set of tools and
so the other guy's job. It's a relay game.
Yeah.
Thank you very, very much, Itay, both
you.
for your research and for the interview.
Thank you.